All right, so I did ROS1 inhibition in non-small cell lung cancer, and then it's just shown as the protein data bank structure of the enzyme. So for the background part of this, the ROS1 receptor is part of multiple pathways like the P13K, MAP kinase, and JAK pathways. These pathways are responsible for cell growth and proliferation and survival. And because if this receptor is like overactive, it's categorized as an oncogene, and we're trying to find the drug that would inhibit all this. So the way we're going to find it is we're going to start with the protein structure and screen a bunch of drugs against the current FDA drug and get docking scores at standard and extra precision. And then we're going to use the hit compounds in an enzyme assay. And then shown in the middle is that is the current FDA drug docked on the mutant, and there's the nitrogen over here with the glutamate residue, which should have which is going to be translated to M, so it's not going to have the charge interaction anymore. And then over here is the docking of the FDA drug on the mutant, like compared to the wild type, they're like overlapped. For the assay, we're going to do this general reaction where ATP and then PTK are going to react to form the first, it's ADP and the first product. And we waited for like 40 minutes for that in wet lab. And then we covered it up and added in the kinase flow and waited an extra 15 minutes before we read it. And then from that, you should be able to generate two curves, one for the wild type, one for the mutant. And you would do this for each of the hit compounds and get your IC50 values. And then this is potentially what you would see. And at the top would be like the current FDA drug. And then you'd be looking for any compounds where the wild type IC50 was less potent and the mutant one was lower. And we said that was compound 3 and compound 5. And then your reference.
Okay. No, better. Ask anytime.
Why is this pathway important?
So this pathway is responsible for just like general cell survival and proliferation, so to keep dividing. So this is the pathway that like when it becomes mutated and overactive, the cells keep dividing and cause cancer.
Okay, so my time. You actually got a very interesting point. You go back to the slide we showed the interaction. Yeah. So this is another usage to do the molecular modeling. Offer the explanation for some observation. So it's shown when you mutate glutamine to a neutral one, then it draw divide drug resistance. But this docking little bit understand because we lost that capture, specific capture. So the counterbalance is one, then there we can change to a hydrophobic group, and you can have the interaction to compensate loss of charge interaction. So in your discussion, in your final report, you can point out this. This is offer the insight for the next development.